this work? Not for a PDF, apparently. Is it this one? Yeah, great. Thanks very much. I'm going to talk about some work that's very much work in progress. When I submitted the abstract for this meeting, I had hoped it might have progressed a bit farther than it has, but that's the way life goes. This is joint work with my former PhD student, Nat. Nat has now finished up and gone back to Thailand, where he has a clinical job, so that's partly why it's been a bit slow to get it finished, but he's left it in the hopefully capable hands of myself and my postdoctoral researcher, Richard Howie, to try and finish up. Now, since this is a Fisher meeting, I thought before moving on to my talk, I would actually just say a few words about my very limited connections with Fisher. So, in fact, my first connection with Fisher is that he does make an appearance in the references of my own PhD DPhil thesis. So there was a couple of papers by him there, or three, I think, including the seminal 1918 paper. However, it has to be said it wasn't a very direct influence on the work in my thesis. Probably the main bit of work that I've done that has been more directly influenced by Fisher's own writings has been on epistasis, gene-gene interaction. In terms of more personal connections, I did my postdoctoral work with Robert Elston at Case Western Reserve University in Cleveland, Ohio. And Robert had actually studied under Fisher when he was an undergraduate at Cambridge. And Robert tells a sort of a slightly ironic story about the fact that when he was coming towards the end of his studies in Cambridge, he was wondering whether he should take the Part 2 in genetics, which would have meant studying with Fisher in a much more direct way. And he asked his tutor for some advice, and his tutor said, the department at Cambridge, Department of Genetics is rather new. This man Fisher is considered to be eccentric by some, and so it may not stand you in good stead in later life for it to be known that you have worked with him. So as a result, Robert chose not to work with Fisher. I don't know that it did him any harm. Robert himself has had a very illustrious career, but it's kind of interesting to see that that was how Fisher was viewed, at least by some people at that time. Finally, the other personal relationship I've had is I was on a ski trip a few years ago, and it turned out that as part of the ski group, there were three gentlemen who turned out to be Fisher's grandchildren. So I was very excited to find that I was skiing with the grandchildren of the great R.A. Fisher. I think I was possibly the only person in the ski group who found that exciting. I think most people didn't really know what it was all about. Okay, so now on to the main subject of the talk. And the kind of motivating project for this is a study that I got involved in a few years ago in an infectious disease known as visceral leishmaniasis, or VL. And we had some data, which was some Brazilian families from two regions of Brazil where essentially the infectious agent is kind of operating everywhere. And so the idea is that everyone's exposed, but some people get the disease, some people don't. And so we were looking for genetic factors that might explain that. We had about 350 families, and they're quite a mixture of small and large families. We did a genome-wide association study, and that, particularly when we meta-analyzed our results together with a equally large Indian case control study of essentially the same disease, identified a single associated locus in the HLA region on chromosome 6, which perhaps is not unexpected for an infectious disease. And for that, we used a standard linear mixed modeling approach with an empirical kinship matrix, which allowed us to model the relationships in our family data and then also to model more distant relationships in the Indian case control data. However, having done the genome-wide association study, we want to additionally apply linkage analysis to the family data to see if that would identify loci that had not been visible through association analysis. Very much the kinds of loci that Sharon was talking about, where maybe you've got perhaps some rare or low-frequency variants that might be on different haplotype backgrounds and therefore wouldn't necessarily show a strong association signal. But we did want to try and use a method that was computationally fast for the fact that we had a number of quite large and complex pedigrees. And ideally, we wanted to allow for possible inaccuracies in the pedigree relationships. So in the association study, using an empirical kinship matrix, you didn't really worry about whether your known pedigree structures were in fact correct or not. You were just using the genetics to kind of model the relatedness. Now, of course, traditionally, the way that people have applied linkage analysis in Mendelian diseases has been to use the technique known as parametric linkage analysis, which has been a very successful technique for localizing genes involved in rare monogenic disorders. And so that relies on having a set of families or pedigrees, each with a number of affected individuals, and you try and look at the co-segregation of the disease phenotype and the alleles at a genetic marker locus under the assumption that the disease phenotype is really reflecting an underlying disease genotype. So the model you've got in your mind is that your pedigrees look a bit like this. You've got a big D disease allele that's segregating down the pedigree, and so the people that get the disease are generally the ones that have the big D allele. The people that don't get the disease are the ones that have the other allele, the little d allele. But what you've actually genotyped is a marker locus or a set of marker loci, and so what you're actually observing is the big A or little a allele that's kind of going down together with the big D or the little d allele. But of course, in linkage, it doesn't have to always be the big A allele. You could have a completely different pedigree where essentially the same sort of thing is happening. But in this case, because the big D allele was on a different haplotype with a little A allele at the marker, then in this pedigree, it's the little A allele that's being transmitted together with the big D allele. So this sort of situation would not be particularly amenable to association analysis in a population-based sense, because there's not going to be an association. So for some families, it's the big A allele. For other families, it's the little a allele that's associated. So in parametric linkage analysis, you don't actually directly test the correlation, but you instead calculate the likelihood of your observed genotype and phenotype data under various assumptions. You then estimate the recombination fraction between either your single marker and your putative disease locus, or possibly between a set of markers and your disease locus, using maximum likelihood techniques. And then you use a likelihood ratio test of the null hypothesis that the recombination fraction between the loci is equal to 1/2. And in genetics, we use the LOD score, which is basically the log to base 10 of the likelihood, the maximized likelihood in terms of the recombination fraction divided by its null hypothesis value. So this is a kind of, traditional standard way of doing LOD score analysis for Mendelian traits. And typically, convincing evidence is taken as a LOD of 3, which corresponds to the data being 1,000 times more likely under the alternative hypothesis that your genotype marker is linked to the disease locus than under the null hypothesis. So parametric linkage analysis was very successful, and indeed is still successful for identifying genes involved in rare monogenic disorders, but it didn't work particularly well for common complex diseases, and that's really why over the last 10 or 12 years it's sort of largely been superseded by association analysis, particularly genome-wide association studies. But in the mid-1990s, there was an attempt to use a different kind of linkage analysis in the hope that that might work better for complex diseases. And that's so-called non-parametric, or sometimes known as model-free linkage analysis. And essentially, it just tries to determine whether members of a family who have similar trait values, so for example, both affected with the disease, tend to inherit genetic material in common from their common ancestors. And it has some advantages over parametric linkage analysis in terms of not having to make so many assumptions. But essentially, you're looking for exactly the same signal. You're sort of saying, in this family, the individuals that are affected have all inherited the same allele in common from their common ancestor at the marker that you've genotyped. And similarly, the individuals in this family have all inherited the same allele in common from their common ancestor. But the actual allele inherited doesn't have to be the same in different families. That's the point. A particular variety of non-parametric linkage analysis is that was used for affected SIB pair studies. So this is where you have really a larger number of small families where you've got two SIBs who are affected with your disease of interest, and you look at what their inheritance is from their parents. So the idea is that you have the SIBs and their parents genotyped, and under standard Mendelian inheritance rules, you would expect that these SIBs to share 2, 1, or 0 alleles IBD with probabilities 1/4, 1/2, 1/4. And so the idea in affected SIB pair studies with non-parametric linkage is just to test for higher than expected proportions of affected SIB pairs sharing 2 or 1 alleles IBD. There are different ways of testing this, but as will hopefully become clear later on, the test that we've really been focused on is one that was introduced by Neil Risch, and that's sometimes known as the MLS, which stands for various different things. It could stand for the maximum LOD score or the maximum likelihood statistic. And in this method, we construct 3 parameters which represent the IBD sharing probabilities for an affected SIB pair at a locus. They have to add up to 1. And we estimate the parameters by using all the genotype data that's in the family, using typically multi-point methods based on hidden Markov models. And essentially, the method comes down to testing the null hypothesis that these probabilities equal a quarter or half a quarter, which is what you'd expect under the null hypothesis, compared to their maximized values, which will hopefully see a skewing towards the higher number of alleles shared IBD. So the MLS method was really designed for SIB pairs. Again, in the kind of mid to late 1990s, these ideas were extended to look at larger pedigrees, and there are different approaches, but in particular, probably the one that's been used the most is one that's due to Kong and Cox. They extended affected SIB pair methods to apply to larger pedigrees. And so the idea in the Kong and Cox approach is to construct a score representing the amount of IBD sharing by affected individuals in a pedigree. There's various ways you can construct the score. You can look at all pairs of individuals, or you can try and look at all individuals simultaneously. And then you construct a likelihood ratio test based on whatever score you've chosen of the null hypothesis that a parameter delta equals 0. And delta is a slightly slippery parameter to define, but essentially it's some kind of parameter that somehow represents the overall magnitude of deviation from null sharing in the family. So the idea is that you can construct this, and this gives you a test that's conceptually a bit like an affected SIB pair test, but it's for larger pedigrees. In order to actually do it, you need to estimate the inheritance vectors in the pedigree, given the pedigree structures and given genotype markers. And what that means is it's typically too computationally intensive for large pedigrees and many markers. So if you want to apply these ideas to larger pedigrees and many markers, you could potentially use Monte Carlo Markov chain approximations. There's various software packages, SimWalk and Morgan, that implement MCMC versions of this kind of approach. Or else, I only recently became aware of a single point approximation known as RAPID, which is not as computation intensive, but because it's single point, it's kind of looking at each marker one at a time, so it's a bit unclear how well that's going to work for SNPs, which are not individually that informative, but that needs a bit of further investigation on my part. And they used a simulation-based evaluation of p-values. Okay, so let's go back to the original starting point of this study, which is that we had these families from the visceral leishmaniasis study, and we wanted to try and apply linkage analysis to them. We wanted to try and use a method that would be fairly fast and potentially allow for possible inaccuracies in the pedigree relationships. So we thought we could actually use an adaptation of the MLS test that Neil Risch had proposed. In fact, we're using an adaptation of an adaptation, which is something that I worked on a few years ago. So I adapted the MLS test a number of years ago to try and model effects at several different disease loci. So that was the focus of these two papers. But along the way, we were also trying to use affected relative pairs of various different types. Now, if you don't worry about the modeling of several different disease loci at the same time, then you just get modeling one locus as a special case of this test. And so for a single locus, the MLS test statistic is basically the sum over the different affected relative pairs you might have in a family. This is all a kind of within family, and then you sum up over families type approach. And then for each one, you sum over all the affected relative pairs, and then you've got this kind of nested sum here where you sum over the probabilities of IBD sharing being 0, 1, or 2. And then we've got these quantities here, where this z hat ij is a population parameter that we're going to estimate via maximum likelihood. That's essentially the probability that an affected relative pair of the same type as type J shares I alleles IBD at a particular genetic position. And then f hat ij is the posterior probability, given the known pedigree structure and the marker data, that pair J share I alleles IBD. And then f ij is the prior probability, given the known pedigree, but not given any genotype data, that pair J share I alleles IBD at the test position. Okay, so as it stands, these quantities here would be calculated, this one here, the prior probability would be calculated based on the known pedigree relationships. This is basically their kind of kinships, or it's related to their kinships. And the posterior probability would be estimated looking at the inheritance down the pedigree, so knowing the pedigree structure and using hidden Markov models to kind of model that. But what we thought was, in recent years, there's been a number of methods and programs that have been developed for estimating pairwise relationships, kinships, through estimating genome-wide IBD sharing. And I guess Sharon talked about one such program earlier on. Essentially, a lot of this has come about because we now have this dense SNP data that's being generated in genome-wide association studies. And in particular, we focused on a couple of programs, PLINK and KING, that will produce estimates of the prior probabilities that a particular pair of individuals shares alleles IBD based purely on their genome-wide SNP data. And this is really mostly for quality control purposes, to check whether individuals are related or not if they're not supposed to be, or also to check that they have the correct relationships. But it can also be used for linear mixed modeling type association methods. So we thought, well, we could just use these estimates, we could just plug them in to our prior probability for the individuals, which would give us some robustness to the fact that we might not know their true relationship, or we might be inaccurate with their true relationship. But remember, we also need the posterior probabilities, these F hats ij. And so we thought, why not just estimate these in the same way as the priors, but using a subset of SNPs in a kind of window of the genome, which is kind of around the position we want to test. So rather than using the full genome, we'll estimate a little, use the SNPs in a little window to estimate the posterior probabilities at a test position. So that's exactly what we do. It's really a very simple approach. So we've called this RIA, which stands for something that I now forget. It's in the title. What is it? Anyway. Regional IBD Association, that's right. Thank you very much. So what we do is we estimate a global expected IBD sharing probabilities using the genome-wide SNP data. So that's kind of, in principle, you could use all of it, or you could do a kind of pruning and thinning of SNPs if you want, which makes it computationally a bit faster. And we plug it into a program like PLINK or KING to do that. And then additionally, for each affected relative pair, we estimate the local IBD sharing probabilities, again using PLINK or KING, but applied to a more limited number of SNPs. So we might just, we just have the SNPs that are in a particular window that we're trying to analyze. But we'll use all the SNPs in that window because it's a smallish window, so we kind of need to use all of them to get as much information as we can. And then we just estimate, we just plug these estimates back into the MLS formula that I showed you previously, which we then maximize with respect to these IBD sharing probabilities, these Zs. Now, internally, we simplify the estimation of the Zs, which is specific for each type of relative pair. So different types of pairs, affected sib pairs, affected cousin pairs, affected uncle, nephew pairs, they would all have their own set of sharing probabilities. We don't really want that. They should be related to one another. So we instead parameterize these Zs in terms of genetic additive and dominance variance parameters, which is possibly the only thing in this talk that's got some connection with Fisher's work on quantitative genetics. So we considered two different variants of RIA. The standard method estimates both of these additive and dominance parameters divided by K, which is just a kind of supposed to be the population prevalence, but essentially it's just a kind of fudge factor. And we also used a no dominance variance version in which we set that parameter to be 0, which in principle we thought might give us a bit more power. But there's kind of key question here, which I haven't really answered yet, which is how do we specify the window size? How do we decide how many SNPs we're going to use to estimate those posterior probabilities? Initially, we based this on a certain number of SNPs. It was something that the user could choose. Now, we think we do rather better by basing it on centimorgan distances, which perhaps makes sense because that is actually more closely related to what we're actually trying to estimate. But I still haven't yet shown you exactly how we choose it. In principle, we could look at different window sizes, and I'll show you the sensitivity to that in a minute. So we had an idea. We wanted to see how it worked in practice, and rather than first of all applying it to the large data set that was the motivating data set, we thought we would apply it to a data set where we knew ahead of time what we were hoping to find. So we thought that would help us see whether this idea even had any merit in it at all. So as a proof of principle, we applied it to a study of a different disease, a kidney disease known as vesicoureteric reflux. So this data set consisted of 555 affected SIB-fair families. So these are much smaller families. You don't need to use a different method for them. You can analyze them using sort of standard methods. And indeed, we had previously used parametric linkage analysis on these families, assuming a dominant model, allowing for heterogeneity, so allowing some of the families to be linked and others not, which generates something called an H-LOD score. And we also used standard non-parametric linkage analysis using the Kong and Cox ZLR statistic that I mentioned previously, implemented in Merlin. So we had about 7,000 SNPs genotyped in about 2,000 individuals. These were selected from a denser set of genome-wide SNPs that the families had been analyzed with. But note that the GWAS analysis that we had initially done using these denser sets of SNPs did not produce any compelling signals. Okay, so here's the results of the standard parametric and non-parametric linkage analysis in these families. This is sort of a bit like a Manhattan plot, I guess, except that what we've got is a LOD score or an H-LOD score or a ZLR LOD score. And what you can see is that these families give, I would say, quite a convincing signal here on chromosome 10 in terms of a linkage signal, and not really anything very much else anywhere else in the genome. If we home in on this linkage signal, so this is the region of chromosome 10 that we are homing in on the bottom, and really the only point of this is to show you that there are a lot of genes in the region. That's kind of what you expect when you find linkage signals. They don't really home in very clearly on what the underlying gene is. So there's still quite a lot of work involved on our part to try and identify, which, if any, of these genes are really driving the linkage signal. Okay, but we thought this was a nice data set that we could kind of see what would happen if we applied RIA to it and how the results from RIA would compare to the results from standard linkage analysis. So this is what we got previously from standard non-parametric linkage analysis. Just go back again. That's basically this plot that I'd shown here. The next plot down is what you get is if you apply the test statistic, the MLS test that RIA uses, but you use the IBD estimates that you got from Merlin. So essentially these are sort of standard IBD estimates from hidden Markov models using the actual inheritance in the pedigree. And so the point of this is really to just compare the test statistics. Is our MLS type test statistic giving similar signals to the ZLR non-parametric test? And I would argue, yes, it is. Again, we've got quite a strong signal here on chromosome 10. We've got maybe some other strong signals that more or less match up with what we got from non-parametric linkage analysis. So they're not identical. They're not identical test statistics, but they certainly seem to be giving you the same kind of inference. What we have below this is the two sets of results you get from RIA. So this is where you actually estimate the IBD sharing using this idea of using a set of SNPs in a window and then just moving the window along the genome and reanalyzing. And I would argue that, again, we see roughly the same sort of signals, although, perhaps a little not quite as clear. Here I'm showing you two different versions. This is where we used 15 centimorgan windows, and this is where we used 20 centimorgan windows. So clearly there's some kind of sensitivity to the window size. And in fact, this shows you the RIA results if we just try different window sizes. So this is 5, 10, 15, 20, 25, and 30 centimorgan window sizes. So you can see that if you have too small a window size, everything really looks very messy. It's quite hard to spot any signals. As you expand out the window size, we would say that visually, when you get, in this particular data set, to about 15 or 20 centimorgans, this signal that we're expecting to see does seem to start coming up. And it stays there even with larger windows. But of course, ideally, you'd like to use as small a window size as you can, because the larger your window size, then the less well you've actually located any finding. And so we'd really want to choose the kind of smallest window size that still gives us a good result. Now, in this case, we kind of know what a good result is, because we had the original analysis. We can analyze it using standard linkage methods. In other data sets, how will we know what's a good result? And that's really something that we still need to explore a bit, whether we could just suggest that you do indeed try a range of different window sizes, and then we use some kind of empirical way of choosing, which is the best window size and what the significance level is, allowing for the fact that you've tried different window sizes. Note, you probably can't see this scale, but the actual test statistics are very different from the different window sizes. So here they're going up to about 20, 15, 12, 10, 10, 10. So it's not just that the signals vary according to your window size, the actual magnitude of the test statistic varies a lot as well. This is a kind of close-up of the signal on chromosome 10, where we really think there's something going on. So this was our results from original non-parametric linkage analysis. This is the peak. This is the results from using the MLS statistic, using the Merlin IBDs. So that finds exactly the same peak. These are the results from RIA with either 15 or 20 centimorgans. In this case, the peak is a little bit to the right. So there's about a 5 centimorgan difference between the position of the signal we get from RIA and the position that we got from standard non-parametric linkage analysis. Given that we're using 15 or 20 centimorgan windows, I don't think that's actually unreasonable. I think we wouldn't really be expecting to be localizing to anything more than about 10 centimorgans anyway. Okay, so what about the real data set that we actually wanted to apply this to? Well, in fact, I haven't shown you the results from the real data set. We did apply it to the real data set. We didn't find anything very interesting. And so this is results from a version of the real data set. So it's basically the same VL genotypes, but we spiked in a simulated effect to see if we could actually find it, if there was actually an effect to be found. So this is our RIA analysis. In this case, we were using Plink rather than King as our kind of engine for doing the IBD sharing. And we had spiked in an effect on chromosome 6 in the same place that we had had a real association, just because we thought we might as well do that. And so we can see that RIA is very successful in finding this, at least kind of visually. That looks like the peak has been found. We compared it to a few other methods. So there's several methods that are available using Monte Carlo Markov chain type methods, because these families are too big. You can't run them through Merlin or another exact program. So we used a suite of tools available in the Morgan package. There's a kind of IBD sharing-based test called LM-IBD tests that's available in Morgan. You can also just use standard parametric linkage analysis, assuming a recessive or dominant or additive model, or indeed assuming the actual model that we had used in our simulation. All of these methods do basically find the same signal. But the IBD test is possibly the least convincing in terms of the signal actually going above the noise. So we were happy that our method does pick up the same signal as these more complicated and computationally expensive methods. However, what I haven't shown you is that if you actually do a standard association test using linear mixed models, actually that also gives the signal. So this is not a situation where linkage is working and association is not. This is a situation where both linkage and association would work. So we've got a little bit of an issue with the test statistic, that the distribution is not very well defined. It seems to be highly dependent on the window size, but not just the window size, but also the SNP density. So depending, what genotyping chip was used, or whether you're using imputed data, that can also alter the distribution of the test statistic. So we do need to explore that a bit further. We do find generally good separation between the distribution of the test statistics under the null and under the alternative in simulations. So for example, these are simulations of the VUR, smaller pedigrees. These are simulations of our big VL study. And essentially, the simulations under the null of the test statistic are in red and green, and the simulations of the test statistic under the effect are in blue. And so you can definitely see that in general, if there's a true effect there, the test statistics will be much larger than when there's not a true effect there. So we think that there's potential promise here, but actually working out what the distribution of the test statistic is in any given data set is something that we're going to need to work a bit harder on. So really that's part of our further work. We do need to investigate potential simulation or permutation-based methods for trying to evaluate the significance of the test statistic. It's not quite as straightforward as I think the example that Sharon gave, and also some of the examples that people have done previously, where they simply permute trait values. That's not going to work very well in our case, because we want to allow for the correlations between the different relative pairs. And we've got affected relative pairs. So if we start permuting trait values, we'll end up with different types of relatives being affected in the pedigree, and so they're not necessarily going to be comparable to our real affecteds in the pedigree. We also would like to have simulations that don't require specification of the pedigree structure, so that rules out the method that's used in the RAPID program, where they actually simulate knowing the pedigree structure. I mean, I guess we could do that, but ideally we'd like our method to not relying on you knowing the pedigree structure. So we're kind of investigating different possibilities. If anyone's got any good ideas, please let me know. One thing we've thought about is a sort of circular kind of permutation, a bit similar to something that Chris Haley's group worked on a few years ago. So we'll see how that works. So we need to try and get a kind of better way of really evaluating the significance of our test statistic. We also need to compare it with alternative approaches. We want to compare it with this rapid approach. It would be nice to compare it with the population-based IBD mapping approaches that Sharon talked about in the previous lecture. I think conceptually they're quite different, because I think those programs are really looking for much more distant IBD, distant ancestry, whereas we're very much looking at a pedigree. We're really just trying to come up with an alternative to standard linkage mapping, which is offering over just a few generations. So I think they're conceptually a bit different, but it would be interesting to compare. And ideally, we need a better simulation, perhaps using some kind of coalescent ideas of a situation where we have got a disease that's caused by rare mutations and on different haplotype backgrounds, which results in allelic heterogeneity that would generate a linkage signal but not an association signal, to see how well our method works in that situation. Okay, so in conclusion, we've developed RIA, which is a new non-parametric test of linkage. It's designed particularly for large pedigrees, but it doesn't require prior knowledge of the pedigree relationships. Only affected individuals need to be genotyped, which is one sort of potentially attractive thing about this approach. And it is computationally simpler and quicker than competing approaches. This is just the kind of time it took to do the analysis of the big VL pedigrees using various variants of RIA and in comparison to the MCMC kind of approaches. It seems to perform competitively with alternative linkage analysis approaches, although certainly for many complex diseases, association analysis like GWAS approaches might be more powerful. But if you are in a situation where there's allelic heterogeneity, which is the situation we think we are in with the VUR data, where we see the strong linkage signal, then we wouldn't expect to see any association signal. Okay, so thanks to my PhD student Nat, who's there, and to Richard, who's there, and who's also in the audience at the back. And thanks to our funders, the Faculty of Medicine in Thailand, who funded Nat's PhD, and the Wellcome Trust, who fund myself and Richard. Thank you.
Well, thank you very much, Helen. That's a nice exposition. Hang on a minute, Walter. I'll let somebody else have a go first.
Well, I have two questions. I mean, the thing that bedeviled SIPEG analysis was heterogeneity of the disease, not of the genetic variant in a particular gene. And that maybe you can overcome by large enough pedigrees. And I noticed in your main example, you had a strong signal on chromosome 11 that persisted alongside the 10. And my first question is that a reflection of heterogeneity? The second question, though, is when you found a significant signal like that, what are you going to do next?
Okay. I'll try and go back to the actual signal, if I can. Well, maybe I can't. I hadn't particularly noticed the chromosome 11 one. We've mostly been focusing on the chromosome 10 one. And for that, we've been trying to do some sequencing in the pedigrees, sort of under the linkage peak, to see if we can find what's driving the linkage signal. So it's a bit different from doing kind of sequencing generally, trying to find a mutation. Really, we're trying to find, are there mutations that are driving the linkage signal, that are being transmitted down, possibly different mutations in different pedigrees. And yeah, I guess heterogeneity is going to be a problem for any method. Again, if you have large enough pedigrees so that in an individual pedigree there's not so much heterogeneity, that would be the hope that would give you more power.
In terms of defining your optimal window, does it matter if it transcends a recombination hotspot?
I think, you could argue that that's going to be adverse for the method, because you're kind of treating a window as if it's essentially not got much recombination, but actually it has. Again, it's a question of if you make the window small enough, then hopefully as you kind of get to the side of the hotspot, then that signal would start coming up more. So I think it's going to be a combination of where any real disease mutation is in relation to the recombination hotspots.
Sharon, did you have any comment back? Because the comment was made on your analysis. I lost you. Oh, yeah, your analysis. Does anyone have any reaction to that? Or? Yeah.
Walter's comment or my comment?
What?
So which comment are you asking about?
I forgot which one it was, but you said, you said it would have been done, it probably come out differently than the analysis that you had done.
We're talking about the difference between the sort of IBD, you know, from long range, long distance relationships compared to sort of much closer relationships.
Yeah. So one thing you can do if you know that you're looking at the close relationships is once you've found the antibody set, you can just keep the longest segments, which are going to be the ones relating to your pedigree that you're interested in. So that part's pretty straightforward. But as you pointed out, the problem about figuring out the genome-wide significance level and not being able to just permute the trait or just simulate a trait, that's definitely an issue. It's something I've been kind of mulling over. It's not an easy problem to solve. Yeah.
No, I'm trying to just get rid of this for the next speaker.
Okay, that's right. Any other questions, comments? Okay, I think Tommy better move for the time. Okay, well, thanks very much. We're all in good time here. The third speaker this morning is Chanda Basu Malik from the Roslin Institute, who's going to talk on a topic I don't think I've ever heard talk on before. And we're sure interested to hear what it is. So it's on the making sense of making sense of GWAS, understanding the genetic basis of human hair shape using mouse models.